Welcome to OncoPharm. I'm your host, John Bazar. I am a professor of pharmacy practice at ETSU's Bill Gatton College of Pharmacy. And this episode is sponsored by Kelly C. PharmD, an oncology education company for pharmacists. Today, we're going to talk about the new approval for Vepdegestrant, which is, and I'm quoting here, a heterobifunctional protein degrader. The degestrant degrader, yeah. A heterobifunctional protein degrader. What does that mean? This is the first drug like this that's been approved. And to kind of orient you if you're listening in the car, on the treadmill, etc. First thing to think about would be bispecific T-cell engagers. This has nothing to do with T-cell engagers, but it's bispecific. So in the bispecific T-cell engager, you have, think of two hands. One hand binds to, say, CD19 on a malignant B cell. One hand binds to CD3 on a T cell. And then the T cell kills the CD19 positive or CD19 expressing cancer cell. So in this, that's the bi, okay? So this has, essentially, it's a neat little molecule that has the bidirectional part of it is, and the heteros, they're different. So one end of this little molecule, think of it as a block, and one end of the molecule is a circle. And let's say the circle binds to the estrogen receptor, okay? We know that it sounds like fulvestrant, and we'll see that it is compared to fulvestrant, which is an estrogen receptor downregulator. And so you have part of this molecule that binds to the estrogen receptor that is in the cytoplasm on the way to the nucleus, presumably to turn on endocrine-driven genes that would then lead to, say, breast cancer progression. This is going to be a breast cancer drug. All right? So one end binds to the estrogen receptor. The other end binds to E3 ligase, which is going to connect ubiquitin to the estrogen receptor. Now, when ubiquitin gets added to a complex or a protein, it is now flagged for destruction by the proteasome, which chews up the proteins. So what Vepdegestrant does is bind to estrogen, binds to E3 ligase. That ligase then adds ubiquitin, and now Vepdegestrant breaks away, but now our estrogen receptor has been ubiquitinated. It's been flagged for destruction. It gets chewed up, and this leads to proteasomal degradation of the estrogen receptor. And then Vepdegestrant can go on and just keep doing this. Vepdegestrant is just like selling tickets to the estrogen receptor death party, all right? Just over and over again. It's like, here, you talk to this guy, you're dead. You talk to this guy, you're dead. Over and over again, that's what happens inside the cell. So the approval here is for ER positive, HER2 negative, ESR1 mutated advanced breast cancer. This is estrogen receptor 1 mutated breast cancer in folks who have already had progression after at least one line of endocrine therapy. This is essentially the same approval of elacestrant and imlunestrant, which are already approved in this space. They are SERDs, selective estrogen receptor downregulators, that have shown PFS benefit versus fulvestrant. Elacestrant and imlunestrant work the same way. There's one warning precaution in the label for elacestrant, which is dyslipidemia, none for imlunestrant. They both are 3A4 substrates and have drug interactions with 3A4 potent inducers and 3A4 inhibitors. Okay, well, Vepdegestrant, as we will see, is a little bit of a different drug. But before we get into this, if you're like a lot of listeners of this pod, you tune in because you're new to oncology, you're trying to get up to speed with the onslaught of new information like this new drug approval, or you want quick weekly updates to keep pace with all this stuff that's coming out, right? If that sounds like you, check out Oncology Insights newsletter, a free weekly email written by fellow OncoPharm Kelly Karlstrom, founder of Kelly C. PharmD and the ELO Collaborative Program. Each week she shares practical pearls, ways to think through oncology concepts without just memorizing guidelines, as well as insights on staying motivated and growing in your practice. It's written by a pharmacist for pharmacists, and it's easy to read. You can sign up in the link in the show notes. All right, so the most practical thing maybe to take away from this, if you're going to be using this drug, as I would counsel a patient, is it's 200 milligrams, it's a tablet, one tablet, once a day with food. That's what the label says. If you look further than the PI, we see that the AUC is increased about threefold, Cmax increased about threefold when taken with a high-fat meal. which must be the justification to take it with food. The label doesn't say to take with a high-fat meal, like say it does for regorafenib. It says to take with food. But the definition of high-fat meal they used in the preclinical work or the early studies were 800 to 1,000 calories per meal that's at least 50% fat. And just if you go to, say, some of your, maybe not favorite, but some of the popular pizza places, 3 large slices of sausage pizza will get you about 1,000 calories, and I'll get you about 45% fat from those calories. So this doesn't say to take with a high-fat meal, but invariably you'll be taking it with you'll get some increased absorption, but that's the reason for taking it with food, is to increase the bioavailability. There is a warning precaution statement for QTC prolongation, so patients do need a baseline EKG, electrolyte to be corrected before starting, and periodic assessment of that. There are, maybe 10% of folks in the study who had QTC prolongation. The average increase in QTC interval was 12 milliseconds, with the upper confidence bound being 15 milliseconds. So it certainly is something that can happen, but is not one of the most worrisome drugs that we have in oncology. And there's also improved fetal toxicity. It is a CYP3A4 substrate, so we do have to worry about strong CYP3A4 inhibitors like voriconazole or 3 and 4 inducers like rifampin. There's also a statement to avoid use with certain P-glycoprotein substrates where minimal increases may lead to serious adverse reactions. They don't give you any guidance on that. They do have the preclinical or the drug interaction study with Vepdegestrant, which is a P-glycoprotein substrate, where its AUC increases twofold. Other P-glycoprotein substrates include apixaban and rivaroxaban. I would count those as drugs with the narrow therapeutic index where you wouldn't want a doubling of the AUC. There's also warning for use with certain UGT1A9 substrates, and we see dapagliflozin's AUC increases twofold in combination with drug. Many of the other SGLT2 inhibitors are also excreted via or metabolized via UGT1A9. So this drug certainly seems to have a more worrisome drug-drug interaction profile than elacestrant or imlunestrant. There's also a decreased bioavailability with PPIs. The AUC goes down by, I think, approximately 84%, and that didn't make the top of the label to avoid PPIs, which is puzzling. It is also looks to be a weak to moderate 3A4 inhibitor, and we see that midazolam's AUC goes up 1.7-fold. So your 1 milligram of Versed is now 1.7 milligrams on average. Rivaroxaban, pretty decent metabolism via CYP3A4, as is apixaban. It's very going to be something to watch to see if we see problems with our DOACs used in this. I would not recommend using them until we have some PK data confirming that we don't have risks and increased concentration with these drugs, which very well may be the case. But with new drugs like this in oncology, we do have these blind spots. As far as overall tolerability and toxicity, we will talk about the pivotal study, which compared Vepdegestrant to fulvestrant, and it's got about 600 people, 300 on each arm. The most common side effect was musculoskeletal pain. We think of that being very bad with aromatase inhibitors. That was 30% with Vepdegestrant versus 23% with fulvestrant. Fatigue was 29% with Vep versus 16% with fulvestrant. Decreased appetite 11% with Vep versus 5% with fulvestrant. Everything else is pretty similar with the exception of the QTC interval prolongation. There was more AST increase with Vep, but similar ALT increase. So it looks to be fairly well tolerated. It is an oral drug that does have the advantage, but the approval here is based off of this the VERITAC study. Just those who are ESR1 mutated. So now we're talking about 130 in each arm, and it's compared to fulvestrant, which we already now have drugs approved for ESR1 mutated disease, so this would not be our standard of care comparison. So what can you really take from the efficacy analysis? Really not a whole lot. We do see an increase in PFS from a median of 5 months versus 2 months. Overall objective response rates was 19% versus 4%. I suppose that's impressive, but I think most folks are going to be looking at least I'm a pharmacist, so I'm going to look at the drug-drug interaction profile more than PFS and cross-trial comparisons. This does look to have the most drug interaction potential of the 3 drugs in this ESR1 mutated space that are FDA approved at this point. This does have a different mechanism of action, though. So folks that are ESR1 mutated and are not having a florid disease progression, they don't have a visceral crisis, we don't know if there's activity in folks who progress on elacestrant with this drug, but it is a different mechanism of action, and that might mean that it still has some activity. Don't know. It's entirely possible that some of fulvestrant's mechanism of action or elacestrant's mechanism of action actually, maybe there's some cross-resistance with resistance mechanisms. We just don't know. But it is a new drug. It is a novel class of drugs and how it works, which is pretty cool. You certainly could imagine more drugs maybe being targeted like this. Certainly proteasome activity, incredibly important, blocking proteasome activity in multiple myeloma, but using proteasome activity to degrade basically transcription factors that are really important for the cancer progression. I would expect to see more drugs approved in this space. So that is what I have for you this week. Thank you so much for listening. You can follow me on X at PharmDnib and on other social media platforms at OncoPharmPod. And until I talk to you again, remember, doses matter.